It feels like we hear more and more about allergies, autoimmune conditions, these chronic inflammatory issues. It really does make you wonder if something fundamental has, well, shifted in our health.
It really has.
Yeah.
And what's becoming clearer and clearer is that a huge part of this puzzle is in that invisible world of microbes all around us and inside us. You could call it an invisible terrain.
Right. You mean bacteria, viruses, fungi, that whole microscopic universe?
Exactly. And in this deep dive, we're going to explore a really compelling idea how our modern urban way of life has profoundly changed this ancient relationship we have with the microbial world. We'll be looking at scientific theories and research that connects our city environments directly to our health.
Okay, so the mission here is understanding how city living impacts our immune systems, right down at that microbial level.
That's it, exactly. And to start, we really need to dig into two foundational concepts, the biodiversity hypothesis and the old friends hypothesis.
Biodiversity. When I hear that, I usually think about, like, rainforests or endangered species. How does that relate to my own health?
Well, the biodiversity hypothesis, which researchers like Tari Hatela and Graham Rook have really championed, suggests that our well-being, especially the balance of our immune system, is deeply tied to our exposure to diverse natural environments.
Okay.
Think of it in maybe two layers.
Mhm.
There's the outer layer, all the rich microbial life in soil, water, plants, animals, and then there's our internal layer, our own community of microbes, gut, skin, airways.
Yeah.
The theory basically says that the outer environmental biodiversity seeds and enriches our inner microbial world.
So, wait, if the environment around me has fewer kinds of microbes, does that directly mean I end up with fewer types inside me?
That's the core idea, yeah. And the hypothesis argues that the big shifts with urbanization, less nature contact, much more sanitation, have kind of put our immune systems at a disadvantage by reducing this vital microbial richness.
Hmm.
It even suggests a potential link between losing large-scale biodiversity, ecosystems vanishing, and the shrinking of this microscopic biodiversity that's so important for us.
That's a huge connection. Is there actual evidence for this, like studies?
There is, yes. Observational studies have definitely shown a correlation. Living in areas with more biodiversity seems to link up with lower rates of allergies.
Interesting.
And for instance, the Finnish allergy program had some promising early results when they shifted focus. Instead of just avoiding allergens, they started encouraging more exposure to nature.
So it's not just about being clean in the traditional sense. It's about encountering a healthy variety of microbes from our surroundings.
That's a great way to put it. And building right on top of this is the old friends hypothesis, which Graham Rook is well known for.
Okay.
This one focuses on a specific group of microbes, the ones we've actually co-evolved with over, well, millennia. These aren't necessarily the common infectious diseases that popped up when humans started living in large groups.
Got it. So not like measles or the flu. What are these old friends then?
They're the microbes we typically acquire from our mothers during birth, breastfeeding from close family, and crucially from the natural environment. Think soil, untreated water sources historically, animals, plants.
Right.
The old friends idea posits that exposure to this diverse set of microorganisms, the ones we grew up with evolutionarily speaking, is absolutely essential for our immune system to develop its regulatory functions properly.
Regulatory functions. What exactly do they do?
Think of them as the immune system's peacekeepers. These old friends seem to play a really key role in stimulating specific cells called regulatory T cells, or TREGs.
Tregs.
Yeah, and TREGs are vital. They stop our immune system from attacking things that are actually harmless, like pollen or dust mites, or even sometimes parts of our own body.
So these old friends basically teach our immune system what not to freak out about.
Exactly. There's this concept called evolved dependence. Our immune systems have, over time, become reliant on these microbial inputs to stay balanced. So the problem in modern urbanized countries might not just be about personal hygiene, but more about a fundamental lack of exposure to these specific immune-regulating microbes we used to get constantly from nature.
Okay, so the biodiversity hypothesis is the big picture. Lots of different microbes, generally good, and old friends zooms in on these specific co-evolved ones that train our immune system's self-control.
Precisely. They really complement each other. You need that rich background of biodiversity to even have the chance to encounter the old friends. And this brings us to a really critical concept, immune tolerance.
Immune tolerance. Yeah, that sounds central to all this.
It absolutely is. It's not just the immune system ignoring stuff. It's an active learning process, learning to tell the difference between a real threat and something harmless.
Okay.
And this learning process relies heavily on getting the right microbial signals, especially early in life, and continuously. When we lack these old friends and live in these microbially depleted environments, our ability to develop proper immune tolerance can be compromised, leading to those overreactions we see in allergies, maybe autoimmunity, other inflammatory conditions. Basically, our immune systems evolved expecting this diverse microbial education, you know? And modern urban life just drastically cuts that schooling short, leaves the immune system sort of undertrained and jumpy.
So it's not just about fighting off pathogens. It's about having a well-educated immune system that knows when to chill out.
Exactly right. We need to shift how we think about immune health. It's not just the absence of infection, it's the presence of these strong regulatory networks shaped by the microbial world. And, well, this really changes how we should think about cleanliness.
Okay, so we've got this crucial link. Microbes shape our immune response. How exactly does urbanization throw a wrench in the works?
Well, urbanization is a really, really rapid departure from those microbe-rich environments where our immune system spent thousands, millions of years adapting. It creates what scientists call an ecological mismatch, or sometimes a gene-environment mismatch.
Meaning our biology, evolved for one world, is now living in a completely different one.
Precisely. Our biology expects a world teeming with microbes, and it suddenly finds itself in environments that are, comparatively speaking, quite sterile.
And the most obvious change is just less contact with nature, right? Fewer farms, forests, less time outdoors.
Absolutely. That's a huge factor. We have way less contact with those primary sources, farms, forests, soil, animals. But it's not just about less nature. Urban lifestyles themselves make it worse.
How so?
Think about the intense focus on hyper-cleanliness, all the sanitation, which, don't get me wrong, is critical for stopping infectious disease spread.
Of course.
But it can also limit our exposure to the broad spectrum of environmental microbes, including the potentially beneficial ones.
And diet must play a role, too. You hear so much about fiber.
Definitely. Modern urban diets often mean lots of processed food, and crucially, low fiber. Fiber is food for many of our beneficial gut bacteria. When they ferment fiber, they produce these things called short-chain fatty acids, SCFAs. They're like messages, powerful anti-inflammatory signals for our immune system. So yeah, a low-fiber diet really messes with our gut microbiome's composition and its ability to help regulate immunity.
Okay. And then there's antibiotics. They seem to be everywhere.
Yes. The widespread use of antibiotics, sometimes maybe indiscriminately used, especially in early life when the microbiome is just getting established, that can have really profound, long-lasting disruptive effects.
Makes sense.
And add to that, medical practice is more common in urban settings. Higher rates of C-sections, for example.
Yeah.
Shorter durations of breastfeeding, often.
We touched on that. C-sections mean the baby misses the mom's vaginal microbes, and
Right.
formula, well, it's not breast milk, microbially speaking.
Exactly. Formula lacks that microbial richness and those special prebiotics, the HMOs, found in breast milk. So you take all these factors together, less nature, hyper-cleanliness, diet changes, antibiotics, altered birth and feeding, it's a major disruption.
A disruption in how we naturally get those old friends and other diverse microbes.
Yes. It leads straight to that immunoregulatory deficit we talked about. Our immune system just doesn't get the training input it evolved to expect.
It really paints a picture. If the training data is missing or corrupted, the system makes mistakes.
Exactly. And we see pretty stark evidence of this environmental impact when we look at, say, immigrant populations.
Oh, how so?
People moving from more rural areas with typically higher microbial exposure into cleaner, urban settings often show a really swift increase in immune-related problems, allergies, asthma, sometimes autoimmune issues.
Wow, that quickly. That's a powerful indicator of the environment's immediate role.
It really is. You could almost view urbanization as this evolutionary bottleneck for our relationship with microbes. By cutting down our exposure so drastically, we're essentially selecting for a less diverse, maybe less helpful human microbiome.
And you mentioned this could even pass down generations.
That's the concerning part, yes.
Yeah.
Reduced microbial exposure in one generation can mean the mother has a less diverse microbiome to pass on.
Right, at birth.
And then urban practices like C-sections, maybe more formula feeding, can further disrupt that already potentially compromised inheritance. It sets up a possible cycle. Some researchers call it the disappearing microbiota hypothesis.
Meaning each generation starts with a weaker microbial foundation.
Potentially, yes, leading to increased susceptibility across generations.
It's quite a thought. So, okay, we're living in these urban settings. What does the actual microbial world look like there? Is it just empty?
Not totally empty, no, but it's definitely a different world compared to, say, a forest floor or a farm. The urban microbial landscape generally has lower overall diversity, a different community structure, and some unique selective pressures.
Lower diversity that keeps coming up. Is that found pretty much everywhere researchers look in cities?
Yes, it's a very consistent finding across many studies,
Huh.
whether you sample the air, the soil, surfaces in cities. The variety of microbes tends to be lower than in more rural or natural spots.
And that affects us directly.
It seems to. This trend extends to us urban dwellers too. Our own microbiomes, gut, skin, airways often show lower diversity compared to people in rural settings. Comparative studies, like in Kazakhstan or comparing urban Chinese populations to rural Mongolians, clearly show this reduced gut microbiome diversity. And it starts early. Urban mothers and their babies often have less diverse microbial communities.
So it's not just what's missing, but also what's there. You mentioned different structure, selective pressures.
Right. Urban environments can sometimes become enriched in certain types of microbes. One big concern is the higher prevalence of antimicrobial resistant strains, AMR strains.
Ah, like superbugs.
Exactly. Especially in healthcare settings within cities. Think MRSA, methicillin-resistant staphylococcus. Studies have found higher rates in urban nursing homes, possibly linked to proximity to hospitals, which can act as reservoirs.
And it's not just in hospitals now, is it? I hear about community-acquired MRSA.
Yes, CAMRSA, spreading in dense urban areas, is another example. It shows these potentially dangerous, hard-to-treat microbes aren't just confined to hospitals anymore. They can thrive in the wider urban environment.
Hmm.
Then you have the selective pressures within our buildings themselves.
Like the materials we use to build. Concrete, steel, glass.
Precisely. Things like concrete, wood, glass, plastics. They create different little niches based on porosity, pH, nutrients, moisture retention. Concrete, for instance, is pretty harsh, high alkalinity, low nutrients, but it still supports specific bacteria that can handle those extreme conditions.
So our buildings are kind of acting like filters, selecting for tough microbes?
In a way, yes. And then think about how we try to keep things clean. Sterilizing surfaces constantly, while important sometimes, definitely reduces overall microbial diversity indoors. It can wipe out beneficial microbes along with the problematic ones.
Okay. What about things like air conditioning? HVAC systems?
Right, HVAC. They filter the air, which is good for removing dust, pollen, some microbes. HEPA filters are very effective. But in filtering out potential harms, they also reduce our exposure to potentially beneficial environmental microbes drifting in. And if they're not maintained well, HVAC systems can become sources of contamination themselves, mold, bacteria.
Oh, right.
Some newer systems even have filters designed to actively neutralize microbes, which further cuts down the microbial load, for better or worse.
So the urban microbiome isn't just a watered-down version of a natural one. It's being actively shaped into something distinct, often less diverse, maybe more antagonistic.
That sums it up well. It's a mix. Lack of exposure to the old friends and other helpful environmental microbes, plus an increased presence of microbes adapted to the artificial environment extremophiles on buildings, AMR strains in dense areas. It creates this unique microbial pressure cooker, contributing to that ecological mismatch.
This really highlights how vital those early years are. How does this changed microbial world affect that critical window for immune system development?
Early life, yeah, from before birth to about age 3, is absolutely foundational. That's when our resident microbes really get established and our immune system goes through major maturation. Dr. Rook used a great analogy.
Oh, yeah.
He said the infant immune system has the hardware and software, but lacks the data. And that data, it comes from microbial exposures.
So how we're born, how we're fed in those first months, that's the initial data download.
Absolutely critical. Birth mode is huge. Vaginal delivery exposes the baby to mom's vaginal and gut microbes, rich in beneficial types like lactobacillus, bifidobacterium, key players in priming the immune system.
Okay.
Contrast that with a C-section. The baby misses that initial wave. It gets colonized primarily by skin microbes, hospital microbes, often results in lower initial diversity, a delay in getting those beneficial bacteria established.
And breastfeeding. We know it's beneficial in so many ways.
It is, microbially too. Breast milk isn't sterile. It contains maternal microbes. Plus, it's packed with prebiotics, those HMOs, human milk oligosaccharides that specifically feed beneficial gut bacteria. And lots of immune factors, too.
So it actively shapes the baby's gut environment.
Actively shapes the gut, shapes immune development, metabolic programming, even influences long-term disease risk. This early microbial ecosystem is incredibly influential.
It sounds like there's an evolutionary script for how this is supposed to happen.
There really seems to be a carefully orchestrated sequence. First, that maternal transfer of vaginal birth, breast milk, that sets the stage. Then, gradually, exposure to a wider range of environmental microbes as the infant grows.
And modern life can interrupt that script.
Significantly, C-sections, antibiotics early on, formula feeding, these deviate from the natural sequence. And we see strong links, associations between these deviations and higher risks of immune and metabolic diseases later. It strongly suggests that timing and the type of microbial succession are absolutely critical.
So it's not just getting microbes, it's getting the right ones in the right order.
Precisely. Now, maybe we should dive into the actual mechanisms. How does this immune education, this microbial dialogue, actually work at the cellular level?
Okay, yeah. Who are the main players in this conversation between microbes and our immune system?
Well, think of key cells, like dendritic cells, regulatory T cells, and then the microbial signals themselves, like PMPs and SCFAs.
Dendritic cells. DCs, what do they do?
DCs are like the sentinels. They hang out at the interfaces with the environment. Gut, skin, lungs, constantly sampling what's there. They're crucial bridges between our innate immunity, the first responders, and the more specific adaptive immune response.
So they're the first immune cells to really see the microbes.
In many cases, yes. And they have these special receptors, pattern recognition receptors, or PRRs. These recognize conserved microbial structures, things called PMPs, pathogen-associated molecular patterns.
The PMPs, like microbial ID tags.
Kind of. Things like LPS from gram-negative bacteria, or peptidoglycans from gram-positive bacteria. Molecules common to microbes but not found in our own cells. When a DC grabs onto a PMP with its PRR, it gets activated.
And what happens when a DC gets activated?
It matures. It starts displaying more antigen-presenting molecules on its surface, ramps up co-stimulatory molecules like Go signals for T cells, and starts producing cytokines, which are signaling molecules.
Okay.
Then the activated DC travels to the lymph nodes and basically presents the microbial bits, the antigens, to T cells. It's like showing them, look what I found.
And that's how the adaptive immune system learns about the microbes.
That's a key part of it. But DCs do something else really important too. They're also critical for guiding the development of those regulatory T cells, the TREGs we mentioned.
Ah, the peacekeepers.
Exactly. And for establishing T cell tolerance, making sure the immune system doesn't attack harmless things.
So DCs aren't just raising the alarm, they're also helping to teach tolerance. That seems crucial.
It is. And TREGs are essential for that balance, preventing overreactions. Some TREGs develop centrally in the thymus, but others, called inducible TREGs or iTREGs, are generated out in the tissues, and their development is heavily influenced by microbial signals and cytokines like IL-10 and TGF-beta.
And that links back to old friends, again, the idea that not getting those microbial signals impairs TREG development.
Absolutely. That's a core part of the hypothesis. Studies in farm environments, which are microbially rich, have shown links between that exposure and higher levels of IL-10, a key anti-inflammatory cytokine that promotes tolerance.
Okay. You mentioned PMPs, the microbial ID tags. What else do they do besides activating DCs?
Well, different PMPs, or different amounts of them, can steer the immune response. Some might push towards a Th1 response, good for fighting viruses inside cells. Others might favor a Th2 response, which is more linked to allergies if it gets out of balance.
Interesting.
And remember LPS, the endotoxin in farm dust. It's strongly linked to protection against asthma and allergies, possibly by triggering an anti-inflammatory protein called A20 in airway cells.
That's fascinating. So something potentially inflammatory in one context can be protective in another?
Exactly. Context and dose are key. And then we have the short-chain fatty acids, SCFAs.
Right, from fiber fermentation in the gut.
Yes. Acetate, propionate, butyrate mainly. These are incredibly potent anti-inflammatory and immune-modulating molecules.
So eating fiber feeds our good gut bacteria, and they make stuff that helps calm down our immune system.
That's a great summary. SCFAs help TREGs develop and function better, they influence what DCs do, and they help keep our gut lining strong. They even have effects beyond the gut. The gut-lung axis is one example. Acetate, for instance, can promote antibody production in the gut.
Which means low-fiber diets, common in urban areas, could directly mess with immune regulation because you're not making enough SCFAs.
That's a really key connection, yes. Less fiber means less SCFA production, which can negatively impact both DCs and TREGs. So if you piece it together, environmental microbes provide the data, PMPs, metabolites like SCFAs. DCs interpret this data and present it. TREGs enforce tolerance based on these signals. If you disrupt any part, low microbial input, skewed DC signals, poor TREG function, low SCFAs, you can get immune dysregulation.
It paints such a clear picture of how vital this microbial input is for a balanced immune system.
Yeah.
So what are the real-world consequences when this system goes wrong, when we don't get enough of the right exposure?
Well, as we've been hinting, this failure of immunoregulation due to inadequate microbial exposure is strongly linked to the rise in all those chronic inflammatory disorders we see so much of in urbanized societies. The common theme, the immune system loses its cool, its ability to tolerate harmless things.
Okay, let's talk specifics. Allergies and asthma. You mentioned the farm effect. How does that fit the microbial story?
Absolutely. The farm effect is incredibly consistent. Kids growing up on traditional farms, especially with livestock, with lots of soil contact, they have significantly lower rates of allergies and asthma.
And that's because
It's largely thought to be because of the richer, more diverse microbial exposures on farms. Things like bacterial endotoxin, LPS, seem particularly important for inducing tolerance in the airways. Studies have shown an inverse link. More microbial diversity in the farm kids' environment and airways lower asthma risk.
and the timing matters, right? That child study you mentioned.
Yes, the Canadian child study by Brett Finley's group was really pivotal. It showed that the gut microbiome composition in just the first few months of life was critical for later asthma risk.
Wow.
Specifically, they found that lacking certain bacteria, the genera Faecalibacterium, Lecnospira, Vianella, and Rothia, sometimes called FLVR at 3-month-old, was linked to a higher chance of asthma later on.
And they could fix it.
In animal models, yes. Reintroducing those specific FLVR microbes actually prevented asthma-like inflammation. It strongly suggests a causal link and maybe a crucial window for intervention.
That is incredible. What about pets? The idea that pets might protect against allergies.
That fits too. Early life exposure to pets, especially dogs, is often associated with reduced allergy and asthma risk. The likely reason. Pets, particularly dogs going in and out, track in a greater diversity of microbes.
Ah, so they diversify the home environment.
Exactly, which likely influences the child's developing microbiome and immune system in a positive way.
Okay. What about autoimmunity, inflammatory bowel disease, IBD? How do they connect to this lack of microbial exposure?
The old friends hypothesis links them directly. The idea is that failed immunoregulation, because we're missing those key co-evolved microbes, leads to problems. Either the immune system mistakenly attacks the body's own tissues, that's autoimmunity, or it overreacts to harmless gut bacteria or food components, leading to IBD. Defective TREG function is thought to be central.
So it's the same basic problem again, an immune system that hasn't learned tolerance properly, misidentifying friend from foe, whether it's pollen or your own cells.
That's the thinking, yes. And the impact might be even broader. There's growing evidence that people in urban settings might have chronically higher levels of low-grade systemic inflammation, even without a specific diagnosed disease.
Low-grade inflammation, I hear that term a lot.
Yes, and it's increasingly seen as a risk factor for a whole host of major non-communicable diseases: type 2 diabetes, obesity, cardiovascular disease, even things like depression.
So it's not just isolated immune diseases, but potentially a system-wide effect impacting overall health.
Precisely. The fact that we see this simultaneous rise in such different types of inflammatory conditions, allergies, autoimmunity, metabolic issues, maybe even some mental health links, suggests there might be a common underlying problem.
A systemic failure of immune regulation.
Driven by our changed relationship with the microbial world, the old friends and biodiversity hypotheses offer this unifying framework. Lack of essential microbes impairs regulatory immune development. And the flip side is hopeful. Interventions restoring these interactions could have really broad benefits.
This makes me think about where we actually spend most of our time. For most of us in industrialized countries, that's indoors. What's the microbial story inside our homes, our offices?
That's such a critical point. People in industrialized nations spend, maybe 90% of their time indoors? So the microbial ecosystems inside our buildings, especially our homes, become a primary source of chronic exposure. And often, at the heart of that indoor world, is household dust.
Dust. That's stuff I'm always trying to get rid of.
Exactly. But it's far from just being inert dirt. Household dust is surprisingly dynamic. It's like a microbial reservoir, a biological signature of indoor life.
How so?
It accumulates microbes from our skin cells shed constantly, from pets, if you have them, from stuff tracked in from
Yeah.
outside on shoes, clothes, even from the building materials themselves. And because it settles and gets kicked back up into the air, we're constantly breathing it, touching surfaces coated in it.
So those dust bunnies are actually tiny ecosystems.
In a sense, yes. Research like Dr. Jack Gilbert's home microbiome project really showed how much we, the occupants, shape our home's microbes. They found when families moved, the microbial signature of the new house quickly started looking like the old one. We carry our microbial fingerprint with us.
That's amazing. So what determines which microbes end up in my dust?
It's a mix. Fungi and dust often reflect the outdoor environment, the local climate. But the bacteria, they tend to be more strongly influenced by the occupants. Number of people, pets or no pets. Even the male-female ratio can leave a bacterial imprint on the dust.
Mm.
And importantly, exposure to these dust microbes is linked to health outcomes, allergies, respiratory issues. So dust isn't just dirt, it's this active biological interface influencing our health.
Okay, so we're living in this indoor microbial cloud. Besides just us humans being there, what else really shapes what's in that cloud?
Well, beyond just us, things like pets, siblings, even indoor plants can significantly change both the diversity and the specific types of microbes indoors and therefore what we're exposed to.
Pets, again, we talked about allergies. How do they change the overall dust microbiome?
Pets, especially dogs that go outside, consistently bump up the bacterial diversity in dust. They're like little microbial delivery systems, bringing in soil and plant microbes from outdoors.
Right.
And there's a lot of microbial sharing between pets and owners. That's probably a big reason why having pets, especially early on, seems protective against allergies and asthma. It diversifies the kid's gut microbiome and just their overall exposure, though the effect might depend on how much time the pet actually spends outdoors.
Makes sense. What about siblings? The classic older sibling brings home germs idea?
They do. And in this context, that might actually be a good thing for the younger siblings' developing immune system. Older siblings definitely impact an infant's early microbiome gut and throat, especially the first year.
How so?
Kids with older siblings tend to have higher gut microbial diversity. And there's even a specific gut microbe signature seen in these kids around age 1 that's linked to a lower risk of asthma by age 6. Yeah, this sibling effect is likely down to just more microbial exchange happening within the family unit.
So a bit of family microbial sharing might actually build a stronger immune system.
Huh?
What about houseplants? Do they do more than just look nice?
They do seem to influence the indoor microbiome. Studies show plants can increase the abundance and diversity of microbes on nearby surfaces. These likely come from the plant's leaves, its phyllosphere, and the soil, the rhizosphere.
Beneficial microbes?
Potentially, yes. They can introduce plant-associated bacteria, though it's fair to say they can also sometimes be a source of fungal spores, which could be allergens for some. But beyond microbes, plants help filter air particles and break down some VOCs, volatile organic compounds.
And things like green walls?
Right, living walls seem to have even bigger effects. Studies have shown increases in beneficial skin microbes on people working near them, and even positive changes in immune markers. So, pets, siblings, plants, they all act as these microbial conduits, diversifying the indoor microbiome, counteracting depletion, maybe giving our immune systems better training.
It's like passive rewilding of our indoor spaces, almost. Okay, let's shift to the buildings themselves, the actual materials and systems. How do they influence the microbes inside?
They have a huge impact. The physical structure, the materials chosen, how we maintain and operate the building, it all shapes the microbial communities that can live there.
Let's start with materials. We talked about concrete being extreme. What about others? Wood, glass, plastics.
Right. Different materials have unique properties: porosity, pH, nutrients, moisture retention. These all select for different microbes. Concrete, like we said, is tough, high pH, low nutrients, but specific bacteria adapt.
What about wood? Seems more natural.
Wood is complex. Some woods have natural antimicrobial properties, but damp wood is great for mold growth. Uncoated wood releases natural VOCs, terpenes, usually low risk. But coatings on wood, they can release different, sometimes more concerning VOCs. Some research suggests things like cross-laminated timber might support lower bacterial loads sometimes.
And glass, plastic, they seem pretty inert.
They can seem inert, but glass can still get microbial biofilms if there's moisture. And plastics, used everywhere indoors, can also provide surfaces for microbes to grow on.
You also mentioned natural plasters earlier, like clay and lime. Are they better microbially?
They offer some interesting properties. Clay plaster is breathable, hygroscopic. It helps buffer humidity, absorbing excess moisture, releasing it when dry. That can reduce mold risk. Some think clay might even absorb certain VOCs.
And lime.
Lime plaster is naturally resistant to mold and bacteria because it's highly alkaline. So yes, these traditional materials can influence the microbial balance differently than, say, standard gypsum board and synthetic paints.
So the materials themselves create specific conditions. What about how we treat those materials, like cleaning?
Cleaning makes a big difference. Frequent use of harsh, broad-spectrum antimicrobial cleaners definitely reduces overall microbial diversity. Good for killing specific pathogens when needed, but it also wipes out harmless or potentially beneficial microbes.
And might select for resistant ones?
Exactly. It might contribute to selecting for antimicrobial resistance. There's even some research linking frequent use of certain cleaning sprays, air fresheners, in homes with young kids to higher rates of wheezing and asthma.
That makes you think twice about spraying everything down constantly. Are there gentler options?
Definitely. Things like vinegar, baking soda can be effective for routine cleaning without the same hit to diversity or the chemical exposure risks. Of course, context matters in a hospital.
Yeah.
In ICU, rigorous disinfection is non-negotiable. It's always a balance.
Okay. And ventilation systems. We know they filter air.
Right. HVAC systems filter out particles, including microbes, reduces exposure to pathogens and allergens, which is good, but also potentially reduces exposure to beneficial environmental microbes from outside.
And poorly maintained ones can be sources themselves.
Yes, they can grow mold and bacteria if not kept clean. And like we mentioned, some advanced filters actively neutralize microbes. Plus, how you ventilate matters. Opening windows' natural ventilation usually brings in a more diverse range of outdoor microbes compared to relying only on recirculated filtered air.
So the whole built environment materials systems maintenance puts strong selective pressure on indoor microbes, often leading to lower diversity, favoring hardy survivors or occupant shed microbes, and often missing those old friends.
That's a good summary. Non-porous synthetics offer fewer niches than natural materials. Material chemistry selects for or against microbes. Biocidal cleaning favors fast colonizers or resistant ones. HEPA filtration isolates us from outdoor microbes. The combined effect. An indoor microbial landscape very different from nature-controlled, depleted, sometimes extreme, contributing to that ecological mismatch and potentially immune issues.
Wow. This has been incredibly detailed. It really begs the question, knowing all this, can we actually do anything to rebuild our immune resilience? What role can things like urban planning, architecture, maybe interventions in early life play?
That is the crucial question, isn't it?
Mm-hmm.
And the good news is the evidence strongly suggests, yes, we can take steps. Urban planning, architectural design, targeted early life interventions, they all have potentially significant roles in reshaping our microbial world for the better.
Okay, let's start big picture. Urban planning.
Yeah.
How can we design cities to be more microbiome friendly?
The main goal here is really aligning with the biodiversity hypothesis. We need to consciously weave microbial diversity into our urban landscapes, our green spaces.
So not just parks, but diverse parks.
Exactly. It's not just about the quantity of green space, though the WHO has guidelines on access, which are important for many health reasons, including immune function. It's also about the ecological quality. We need to move beyond just neatly manicured lawns towards creating more complex ecosystems within the city. These can support a richer, more diverse aerobiome, the microbes floating in the air. This means things like promoting biodiverse plantings, using native species in parks, along streets, in gardens, on green roofs. This creates better habitats for beneficial soil and plant bacteria. Some cities, like Breda in the Netherlands, are already doing this.
What about rewilding? I hear that term.
Yes, rewilding parts of urban areas, allowing more natural development with less intensive management, can boost ecological complexity and microbial diversity. Even our infrastructure can help. Green infrastructure for stormwater, like rain gardens or bioswales, can host unique microbial communities.
And what about places kids spend a lot of time, like schoolyards?
Transforming schoolyards is a huge opportunity. Replacing asphalt and rubber surfaces with natural materials, soil, plants, rocks, even forest floor litter. Studies, especially from Finland, show really positive effects. Changes in kids' skin and gut microbes, increases in those important regulatory immune markers, potentially lowering their risk of immune diseases later. Their microbial profiles start looking more like kids who have regular forest contact. Even just having natural rocks versus rubber mats on playgrounds makes a difference in the microbial communities found there.
That's concrete.
Yeah, And there are research projects now, like Impetus UG Micro, specifically tracking how microbes transfer from these urban green spaces to people and the health effects.
So the key for urban greening, from this perspective, is focusing on ecological function, maximizing microbial biodiversity, not just adding green aesthetically.
Precisely. It has to be about creating and maintaining green spaces that are vibrant, functioning ecosystems, delivering those crucial microbial benefits. Remember, contact with natural environments enriches the microbiome. Ecological quality is paramount. We need urban planning and landscape architecture to see green space as vital ecological infrastructure for health.
Okay, that covers the city outside. What about bringing this thinking inside our buildings?
This is where the idea of probiotic architecture emerges. It's really a paradigm shift, thinking about designing buildings to intentionally incorporate and foster beneficial microbial communities.
Probiotic architecture. I like the sound of that. What does it involve practically?
It looks at several things. Material choice is big. Using natural materials like clay and lime plasters, they breathe, manage humidity, potentially reducing mold, creating a more stable microbial environment. Clay might even absorb some pollutants.
Wood, too?
Wood, managed properly for moisture, yes. Then there are green walls, living walls. We know they can increase beneficial skin microbes, filter VOCs, maybe positively modulate immune molders.
What else?
There's research into bioreceptive materials designed specifically to host beneficial microbes you might intentionally introduce. And ventilation is key. Prioritizing natural ventilation, operable windows brings in more diverse outdoor air. Even the building's shape can influence air and microbe flow.
Is anyone actually testing this? You mentioned a school study.
Yes. That study looked at interventions in a school. They found the biggest drop in problematic microbes, and AMR happened with a combo of better ventilation and using probiotic-based sanitation cleaning with beneficial microbes to outcompete bad ones.
Interesting.
So probiotic architecture isn't one single thing. It's a holistic approach.
Yeah.
Smart material choices, design that works with natural processes like airflow and humidity, maybe even targeted microbial seeding. It's moving from trying to eliminate all microbes to cultivating the right ones.
This is fascinating at the city and building scale. But what about more immediate actions? Targeted things we can do in homes, schools, healthcare settings, especially for young kids in that critical window.
Absolutely. That's where targeted early life interventions come in. In our homes, it starts with rethinking some parenting advice and cleaning habits.
Like easing up on the harsh cleaners.
Exactly. Reducing overuse of strong antimicrobials that wipe out diversity, opting for milder, natural cleaners for routine stuff, emphasizing that super sterile isn't the goal, some everyday microbial exposure is good. Reading labels, maybe choosing unscented products to cut down on VOCs.
What else at home?
Promoting beneficial transfer. Supporting breastfeeding. Considering pets, especially dogs. Recognizing the microbial mixing that happens with siblings. And family diet, plenty of fiber to feed those gut microbes and make SCFAs. Good ventilation helps too, bringing in fresh outdoor air.
Okay. What about schools and daycares?
Playgrounds are key there, swapping artificial surfaces for natural ones, soil, plants, rocks, forest litter. We know this impacts kids' microbes and immune markers positively. Indoors, plants, good ventilation, maybe considering probiotic cleaning methods. And hygiene, essential hand washing, yes, but minimizing routine use of harsh disinfectants, especially when kids are present.
And what about healthcare settings, especially neonatal units where infection control is so critical?
That's the most challenging setting, obviously. Infection control is absolutely paramount in NICUs. But there's growing awareness of the neonatal microbiome's importance.
So how do you balance that?
Carefully. Exploring things like vaginal seeding post-C-section, maybe administering specific probiotics, especially for preemies. Strongly supporting breastfeeding. Even looking at NICU design and cleaning protocols. How can we reduce pathogens effectively while maybe finding ways to support beneficial commensals where safe, like bathing practices or antibiotic stewardship?
Using new tools, maybe?
Yes, advanced tools like DNA sequencing for microbial surveillance, tracking pathogens, AMR, checking cleaning effectiveness, and forming targeted strategies. It requires a really nuanced, context-specific approach. A gradient of microbial exposure, different levels appropriate for different settings, and individual vulnerability.
So more nature contact and less aggressive cleaning at home and school, but super stringent hygiene in NICUs, while still trying to support good microbes where possible.
That's the general idea. Balancing necessary hygiene with beneficial exposure.
This has been incredibly insightful, a real deep dive into this invisible world. To wrap things up, what are the main takeaways and where do we go from here?
I think the core message is this. Our immune health is fundamentally tied to the microbial diversity around us. Urbanization has significantly disrupted this link, contributing to the rise in immune-related diseases.
But it's not hopeless.
Not at all. That's the encouraging part. We can take conscious steps to rebuild these relationships. Now, looking ahead, there are still gaps research needs.
Like what?
We need to pinpoint more old friends, understand exactly how they work. We need long-term studies of interventions, dose-response relationships, how much exposure is needed. We need more validation for probiotic architecture, direct links between building design and health outcomes, and understanding how building features and occupant behavior interact.
And challenge.
Definitely challenges. Microbial complexity telling good from bad isn't always simple. Public perception overcoming germophobia, implementing big changes in city planning, building codes, ensuring everyone has access to these healthier environments, equity.
It requires a real shift in thinking.
It does, a paradigm shift. Seeing microbes not just as enemies, but as essential partners. It leads to this idea of microbial stewardship, consciously managing these invisible ecosystems for mutual benefit, like we're learning to do for forests or oceans.
Microbial stewardship. I like that.
By understanding urban and indoor microbial ecology, applying these co-evolutionary principles, we really can design healthier cities, healthier buildings, adopt lifestyles that support this partnership. It needs collaboration architects, ecologists, immunologists, planners, public health folks.
A huge undertaking.
Huge.
Yeah.
But the potential payoff for future generations' health, for our relationship with the microbial world, immense. It all depends on us successfully navigating and reshaping this invisible terrain.
So the big takeaway for me is, yes, our modern world changed our microbial exposures, impacting immunity. But, crucially, we have the knowledge and potentially the tools to consciously rebuild those connections.
Exactly. And maybe for you, listening right now, this deep dive makes you think about your own daily microbial world. Are there small changes you could make in your home, your community? Thinking about that balance between cleanliness and maybe welcoming a bit more beneficial microbial richness.
Perhaps it's time to embrace a few more of the right kind of microbes. If you're interested in digging deeper, you could look into local urban greening projects, research on natural building materials, or find resources on supporting a healthy home microbiome. It's a fascinating, vital area of ongoing discovery.